Now that we've learned about decapitation studies and the animal studies, let's take a look at the nerve cell studies that were conducted in the 1800s and 1900s. The first microscope was invented in 1590 by a father-son duo, and researchers could see bacteria as early as 1675. Over the next 200 years, it would go through several revisions. You can see the 1847 version here on the screen. This is actually Charles Darwin's microscope that he used in the 1840s. These technological advances allowed researchers to see things that they had never seen before, including nerve cells. Camillo Golgi was an Italian pathologist who developed a special staining technique using silver nitrate. This technique allowed researchers to look at nerve cells under the microscope. In the image on the screen, you can see Golgi's actual illustration of the brain cells that he was able to see under the microscope. At about the same time, a Spanish pathologist and neuroscientist, Santiago Ramon y Cajal, suggested that there must be a space between these nerve cells. He also looked at them under the microscope. In 1906, they actually shared the Nobel Prize for establishing the neuron as the basic unit of the nervous system. Here you can see that Cajal's illustration of nerve cells is a little bit more detailed than Golgi's. Sir Charles Scott Sherrington was an English neurophysiologist. He was focused specifically on the functions of the mind, and he approached the study of this through physiology. He studied spinal dogs, dogs that had had some type of damage done to their spinal cords. Their reflexes tended to be more pronounced than those of intact dogs. So he concluded that the cortex had an inhibitory effect on reflexes. When the cortex was damaged in some way, those inhibitions were raised, and dogs were more likely to spaz out whenever they should simply just turn their head or lift their ears. Sherrington published a book in 1906 to explain how the nervous system and the body coordinate to make the behaviors that we see every day. Reflexes, he said, are the simplest unit of this integration. Complex behaviors then are the result of these reflexes, these simple behaviors chained together, something he called reflex chaining. The synaptine, as he called it, we call it the synapse today. The synaptine is the site where these basic reflexes interact, join together, chain together to create complex behavior. Sherrington also proposed the idea of neurotransmitters, some chemical that allows neurons to communicate with one another. In 1932, he won a Nobel Prize with Edgar Adrian for their work on the functions of neurons. In this lecture, we reviewed some of the earliest studies looking at decapitated heads, animal brains, and nerve cells. All of these studies contributed to the emergence of psychology in the 1800s. In next week's lecture, we learn about German psychology and the father of modern psychology,